Light is all around us. Sunlight drives all natural life on Earth, facilitating everything from photosynthesis to organism populations. Over the past few centuries, light has fascinated scientists all over the world. Its bizarre characteristics and interesting dual nature give light the ability to do things scientists had previously thought impossible. Even the path of light itself is interesting. From our sun, light travels approximately 149.6 million kilometers to reach the Earth. It does this in roughly 8 minutes and 36 seconds at a whopping speed of 300 million meters per second, making it one of the single fastest things in existence. So why is light so important? Well, because of fields like optogenetics, which can make use of light's unique and fascinating abilities, we have a better world today.
Electricity is one of science's biggest mysteries. It is a form of transferred energy that can be generated by anything from the wind to water to sunlight. Things like heating on a cold day and the light bulbs all around us are powered by the mysterious nature of electrical energy. Lightning is an example of electrical discharge in which the bolt can travel at speeds of up to 130,000 miles and reach temperatures of up to 29,982 degrees Celsius. Now onto genetics, a key field in the future of medical research. Genetics dictates how characteristics are obtained and passed on to future generations. It controls everything from our ability to see certain colors to our chance of being affected by certain diseases. On this episode of the Beyond Tomorrow, we will be talking about the field of optogenetics with Dr. Kiran Banerjee. Kiran, what can you tell us about optogenetics?
Well, you see, Vishal, optogenetics is the fusion of optical and genetic sciences. The former is the scientific study of sight and the transmission of light, while the latter relates to how biocharacteristics are passed down through reproduction.
So what exactly does optogenetics entail?
You see, Vishal, it is the use of light to control cells, in particular neurons, within the brain.
Dr. Banerjee, has optogenetics been around for a long time or is it a newly founded field? What can you tell us about the history of optogenetics?
Initially perceived as a far-fetched and ridiculous idea, the idea of using light to control specific individual neurons within the brains was actually thought of by Francis Crick in 1999.
As an interesting fact for our viewers, let me note that Francis Crick is notable for discovering the double helix structure of DNA along with James Watson. Kiran, talk about some of the experiments that have been conducted over the years in relation to optogenetics.
Well, in one experiment, researchers used the light to remove memories of fear within mice and replace them with positive memories. It would be hard to use such a process on humans due to ethical concerns. However, there's a huge potential for such methods could be used to affect and treat the effects of trauma.
Dr. Banerjee, I am, and I'm sure our viewers are as well, curious to know, why optogenetics? Why not another typical medicine field, such as dentistry?
Optogenetics interests me, as it is a field with so much potential to do good for the lives of others. In the future, this particular science could be used to treat conditions such as depression, chronic pain. In fact, a company based in Michigan called the RetroSense is utilizing optogenetics to attempt to treat a genetic disorder leading to blindness. The words of the neuroscientist Robert Giroux from Washington University in St. Louis said optogenetics allows us to do experiments that were not doable before. This is a true game changer like few other techniques in science.
Apart from people with the conditions you've mentioned above, who else will impact from progress in optogenetics?
It's a good question, Vishal. But progress will have implications for the wider medical community as well. Pharmaceutical companies are a good example. This is because optogenetics allows certain cells to be turned on and off. Just think of it as a traffic light. Orange allows the cells to be turned off and green on. This then allows scientists to determine the exact neurons which contribute to particular diseases and allow them to subsequently develop drugs which treat these diseases. It is important to note that narrowing the diseases down to specific circuits within the brain is vital in producing effective drugs to treat them.
So what do we need to know about the risks and issues associated with optogenetics, and what needs to be done in the future?
Like any field, optogenetics has its shortcomings. There are definite issues that we must look to solve. Firstly, we must accept that the procedure is fairly invasive and does include connecting a light source that is very rigid to the human brain. It would likely work quickly with regular human activity. Just look at my experiments with mice. This technology is good in lab conditions, but whether it is viable in regular active humans, is a different question. But down in Justin Gammore's lab, I know that they're currently developing a more flexible and malleable wiring that may better suit the cause. We are likely to progress onto human trials within the year. Secondly, not all cells are created equal. What I mean by this is that different cells exhibit different levels of response at different levels of stimulus, meaning that a lone stimulus, like a single light source, may have different effects on different cells on the basis of location and cellular structure. This phenomenon is known as selective expression and is slightly more difficult to tackle. I think that this is something that the optogenetic community is discussing heavily at the moment.
Are there any other issues other than those that are science related?
Now, these are merely biological issues. Any seasoned medical researcher knows that they only act as secondary concerns when confronted with legal issues. Legally, gene therapy is completely illicit in the United States, with only one legally verified program running in Europe. It is sad for me to see that we cannot put aside our personal beliefs for the betterment of millions of lives. However, I can only hope for the future.
What are 3 important things that still need to be found out?
Gee, Vishal, you're asking some tough questions today. Well, firstly, we need to document specifically and accurately the exact effects that this has on human beings. It is important that we make the most use of our trials and research extensively before bringing any form of this procedure or related substances to market. We need to be sure that animal testing is compatible. Secondly, we need to consider whether this will just allow money to create a bigger divide. What I mean by this is, how can we be sure that wealthy individuals won't use optogenetics to enhance their memory? It is our duty to promote equality and opportunity, and we believe that this is just another means in which that duty may be jeopardized. Thirdly, technological drawbacks may mean that we need to find quicker GPU devices and cloud computing to ensure the success of our devices in the long term.
Being such a high achiever, what is one thing that you would like to share with us that helps to keep you motivated every single day?
It's a good question, Vishal. But I think you forgot to consider one thing. The only time success comes before work is in the dictionary.
In the following segment, I will be going into more depth regarding optogenetics, looking at relevant diagrams for example. But before doing so, I must remember to credit the Universitat Autònoma de Barcelona for writing an article which formed a large quantity of the basis of Dr. Kiran Banerjee's discussion today. Without further ado, we shall observe relevant diagrams of optogenetics. The diagram you can see at the moment summarizes how optogenetics actually works. Neurons are electrical devices, and optogenetics involves using light to manipulate the complex array of electrical circuits within the human brain. However, prior to this, something comparable to a solar panel must be embedded within the desired neurons, which allows light to be converted into electricity. The left-hand side of the diagram depicts a particular algae, and its membrane is home to numerous little proteins called channel rhodopsins, which can be used for exactly the process detailed earlier. These proteins act upon the emittance of blue light, which allows a little hole to open and thus charged particles to enter and result in having an electrical signal. Then, the second and third steps of the diagram show how these molecules are inserted into neurons of the brain. Since the channel rhodopsins are proteins, this means that they are in the DNA of the algae in question. Therefore, this DNA is removed and put into a gene therapy vector, which is basically the means by which the genetic material can be transferred, an example being a virus. The neurons, which create proteins naturally, help to spread these light-conducive channel rhodopsins where the virus is implemented. Subsequently, certain neurons would be able to be controlled by firing and using blue light to open and close ion channels. Nearby neurons, which don't have the molecule, would be unaffected. In this second diagram, we are shown a visual representation of how optogenetics has been used in experiments involving mice. The left-hand side shows thin optrodes, or fiber optic cables and electrodes, being put into the mouse's brain. Moving on to the right-hand side of the diagram, it shows that one of the cables allows blue light to enter the brain and trigger the relevant genetically modified neurons, and thus make a specialized protein. This is basically the on switch. The other cable allows red light to enter the brain and silence other neurons that have been genetically modified using a different protein. This is the off switch in the circuit. In the complex network, if a neuron is activated by a light, it can then send signals to connected neurons. Thank you all for tuning into tonight's episode of The Beyond Tomorrow. Particular mention to this week's special guest, Dr. Kiran Banerjee. We hope you learned something of value about the field of optogenetics.
Back next Thursday at 7 p.m.